Your exams don't do flap, neither do we. Straight talk without the sweet talk. All you have to do is listen, read, test, and destroy. Let's start this lecture now.
Now let's break down the microbiology stains on exams, why gram-positive bugs stay purple, why gram-negatives turn pink, and why tricky organisms like Mycobacteria or Chlamydia play hide-and-seek under the microscope. By the end of this video, you'll not only know the colors, you'll understand the why, the how it relates to virulence, and the clinical implications for antibiotics and immune response. Stick around, because these are the concepts the boards loves to test in subtle ways. Let's get started. Gram staining is the first-line laboratory test for classifying bacteria based on cell wall composition. Gram-positive bacteria have a thick peptidoglycan layer with extensive cross-linking, which traps the crystal violet iodine complex during the decolorization step, so they appear purple. Gram-negative bacteria, in contrast, have a thin peptidoglycan layer located in the periplasmic space beneath an outer membrane. During the alcohol decolorization step, the outer membrane is dissolved, and the thin peptidoglycan cannot retain the crystal violet, allowing the counterstain safranine to impart a red or pink color. These structural differences explain both the staining pattern and susceptibility to antibiotics, as gram-negative bacteria are more protected by their outer membrane, whereas gram-positive bacteria are more directly accessible to beta-lactams and glycopeptides. Some bacteria do not stain reliably with gram stain due to unique structural features. Spirochetes such as Treponema and Leptospira are too thin to be visualized under light microscopy. Mycobacteria have cell walls rich in mycolic acids, creating a waxy, lipid-heavy barrier that resists standard gram staining. They require acid-fast stains to be seen. Mycoplasma and ureaplasma lack a cell wall entirely, so gram stain is ineffective. Certain intracellular pathogens, including Chlamydia, Rickettsia, Legionella, Bartonella, Anaplasma, and Ehrlichia, either lack a typical peptidoglycan structure or live primarily inside host cells, making gram staining unreliable. Recognizing these organisms in clinical vignettes often requires alternative staining methods or serologic/antigen detection. Giemsa stain binds nucleic acids and is used for bacteria with intracellular lifestyles or for protozoa. It is particularly useful for visualizing Chlamydia, Rickettsia, Borrelia, H. pylori, and protozoa such as Plasmodium or trypanosomes. The effectiveness of this stain relies on the presence of dense nucleic acids in host or bacterial cells, which interact with the basic and acidic dyes in the stain. Clinically, Giemsa staining allows visualization of organisms that may otherwise be missed due to their small size, intracellular location, or lack of peptidoglycan. Periodic acid is Schiff-PS. Stain identifies carbohydrates such as glycogen and mucopolysaccharides, which are components of certain bacterial walls or the intracellular inclusions they produce. It is classically used to diagnose Trophorrima whipplei, the agent of Whipple disease, because PAS binds the glycoprotein-rich bacterial remnants accumulated in macrophages. This highlights how structural composition determines staining behavior and allows targeted diagnosis. Acid-fast stains, such as Ziehl-Neelsen, carbofuchsin, detect bacteria with mycolic acid-rich, lipid-heavy cell walls, notably Mycobacteria and partially acid-fast Nocardia. The high lipid content resists decolorization with acid alcohol, allowing the stain to remain within the organism while background cells are decolorized. Oramine rhodamine fluorescent stains exploit the same lipid binding principle but increase sensitivity, allowing rapid screening. The acid-fast property also correlates with slow growth and resistance to common antibiotics, which is often tested in board questions. India ink and mucicarmine stains target the polysaccharide capsule of Cryptococcus neoformans, a key virulence factor. India ink creates a negative contrast, revealing the capsule as a clear halo, while mucicarmine stains the capsule red. The capsule prevents phagocytosis, so visualizing it directly connects structural composition with pathogenicity and helps explain susceptibility in immunocompromised hosts. Silver stains highlight organisms and fungi with cell wall or extracellular components that bind silver ions, such as H. pylori, Legionella, Bartonella henseli, Pneumocystis jirovecii, Aspergillus, and coccidioids. The high affinity binding to cell wall polysaccharides or surface proteins produces black deposits, making these thin or intracellular organisms visible. Fluorescent antibody stains work on a similar principle by targeting antigenic components with labeled antibodies, allowing identification of bacteria, viruses, and protozoa such as Giardia, Cryptosporidium, and Pneumocystis, and are often used in confirmatory diagnostics. Overall, understanding bacterial staining depends on linking the chemical composition of the cell wall or capsule to the interaction with specific dyes, recognizing structural limitations that prevent visualization, and connecting these features to clinical context, virulence, and treatment implications. This approach aligns with board expectations, which often test the reasoning behind staining results, not just memorized colors. Some bacteria demonstrate partial or weak gram staining, which can create diagnostic confusion. For example, Nocardia is partially acid-fast due to intermediate mycolic acid content, and certain fastidious gram-negative organisms may stain inconsistently because of thin peptidoglycan or delicate outer membranes. Recognizing these subtle deviations is critical in clinical vignettes where the description of staining may be atypical rather than explicitly stated. Clinical correlations further reinforce the relevance of staining patterns. Giemsa stain is particularly useful for intracellular infections, such as Chlamydia, Rickettsia, and Plasmodium, because it highlights nucleic acids concentrated within host cells. Periodic acid is Schiff-PS. Stain is classically used to detect Tropharima whipplei within macrophages in Whipple disease, linking bacterial carbohydrate-rich components to disease pathology. Acid-fast staining, applied to Mycobacteria, not only allows identification but also signals the need for specialized treatment regimens, as these organisms' lipid-rich walls confer natural resistance to many standard antibiotics. From a board's perspective, staining is more than just a visual tool. It guides antibiotic selection and predicts immune evasion strategies. Gram-positive organisms with exposed peptidoglycan are typically accessible to beta-lactams and vancomycin, whereas gram-negative organisms, protected by an outer membrane and periplasmic beta-lactamases, often require drugs capable of crossing porins or resisting enzymatic degradation. Similarly, intracellular pathogens highlighted by stains like Giemsa or PS are less susceptible to extracellular immune defenses and may require antibiotics that achieve intracellular penetration. Understanding these connections between structure, staining, and therapy allows exam takers to reason through complex board vignettes rather than relying solely on memorized tables.